Hi and welcome to this video about genes and genetic testing, and we're going to be looking at some exam questions. And so these exam questions, they're taken from the Edexcel AS Biology course, and we're particularly looking at Unit 1 today. And so this question, cystic fibrosis is a genetic disease that can affect many body systems including the respiratory system. Explain how the gene mutation causes a buildup of mucus in the respiratory system of a person with cystic fibrosis. And so you have to explain how you get the mutation within the person, and then how this causes the symptoms of cystic fibrosis. And so the first thing to mention is, due to the mutation, obviously you have to refer to the CFTR protein, and so the mutation means that the CFTR protein cannot be made properly, and therefore you have a different sequence of amino acids. From there, obviously, this means there's a change in the protein, so you could mention a change in the tertiary structure of the protein. You have to mention what the CFTR protein does, and it's of course involved in moving chloride ions out of the cell. And so what this means is, if it's non-operational and it doesn't work and it's the wrong shape, therefore the chloride ions don't move out of the cell and go into the mucus. What this means, also another thing to reference, is obviously the sodium ions are then moving. There's another channel that moves sodium ions into these epithelial cells, and so those sodium ions are moving in. And so what we're thinking about here is a build-up of these ions found within these epithelial cells. And what that means is the water doesn't move out of the cells into the mucus, and hence making it quite thick and sticky. Obviously you can then talk about how the water moves, and that process of course is osmosis. This means that it becomes thicker because the water isn't moving out, and obviously this is related to the fact that it can't be moved, and therefore you get an increase in coughing and those similar types of symptoms. And from this, it's any five which are acceptable there. And so the following question, the one that asks for the definition for these terms. So mutation, this one's worth 2 marks. So it's a change in the DNA, first of all, and you have to therefore mention that it could basically result in a change in the base sequence of the DNA, and that gets you the 2nd mark. Recessive, that refers to the idea that both alleles need to be present in order to have the condition or the recessive phenotype. So an example of that might be cystic fibrosis. In order to have it, you need both alleles in order to have the disease. If it's dominant, therefore, you only need one allele for the dominant phenotype to be expressed. So an example of a genetic condition is Huntington's. For that, you only need one of the alleles in order to have it. Okay, so this next question. Phenylketonuria is a recessive genetic disorder characterised by a lack of the enzyme phenylalanine hydroxylase. When there is an excess of phenylalanine in the diet, this enzyme converts the amino acid phenylalanine into the amino acid tyrosine. Okay, so part B, the number of investigations have been carried out into the use of somatic gene therapy to treat mice that have phenylketonuria. The graph below shows the results of one such investigation. And so you've got the control group, and it shows the concentration of phenylalanine in the blood, and the mice given the gene therapy. And so just by looking at it at a glance, you can clearly see the mice who've had the gene therapy have a much lower level of phenylalanine in the blood compared with the control group. But the first question, use the graph to describe the effect that gene therapy has on the phenylalanine concentration in the blood of these mice. And so it's simply a describe question, and so therefore you had to describe what's on the line. So, and it's only looking at the mice with gene therapy. So in this instance, you describe that there's a steep drop in the first 7 days, and then it remains reasonably constant from there on in. So there you go. So there's a drop in the first 7 days, and therefore it then stays the same. It reasonably levels out in the rest of that time period. Part 2, describe how some somatic gene therapy may have been carried out to treat the phenylketonuria in these mice. So, with the gene therapy, what you're doing is you're introducing the healthy allele or the healthy gene into an organism that doesn't have the healthy allele. And so obviously you need to get that healthy allele into the mice in this instance. And so if you refer to them using the normal or healthy allele or gene, that gains you a mark. From there, you have to mention that that allele codes for phenylalanine hydroxylase, or the enzyme that does the conversion. You need to mention that. And therefore, you'd have to reference how that's introduced into the target cells. From there, you then have to say that that's targeting into, it's going to be put into the DNA, into the chromosomes, or into the nucleus, so mentioning put into some genetic material. And from there, you reference the use of a vector, so how it gets from how that section of DNA is placed into the organism, and so we use the word vector, so transport system. And then you can mention the type of vectors used, so for example a virus or a liposome. And you could also therefore credit the use of the delivery method, so for instance as a spray maybe, or as an injection there. What you'll note about this question that you might have seen in some of the textbooks is it doesn't ask you about specifics about the enzymes used, so for instance ligase or restriction endonucleases to cut the specific gene out. It wouldn't hurt to put that in your answer, but on this mark scheme you don't need to include it. And part 3 suggests what treatment the control group of mice may have been given in this investigation. And so, as it's a control, what you want is essentially something, again they're treated in exactly the same way, so if they're given a nebulizer, or if they're given an injection, then essentially you give them the injection that's empty, that only has, for instance if you used a virus, it's only got the virus in it but not the genetic material, or if you've used liposomes, then empty liposomes, or a water or a saline solution in an injection.